PARASITE Project started in 2013 with the aim of assessing and addressing the risk posed by nematode parasites in the European fishery products, all along the production value chains, tackling the main research gaps identified by the European Food Safety Authority to this respect. It involves 21 partners from 10 European and 3 Asian countries, which have worked together to provide new scientific evidence and technological developments to detect, monitor, and mitigate the impacts of parasites that can be naturally transmitted to humans, that occurs in European caught and imported fishery products. Industrial involvement has been crucial in addressing the protocols.
The fishery sector is really concerned about the negative effects that fish parasites cause and might be able to cause. These effects, when passing onto the public opinion in a not very precise or sensationalist way, may lead to very negative consequences in the market, lowering prices and demand. This is the reason why our cooperative has taken an active part in this project, in order to contribute to an accurate knowledge of the parasites and their consequences, and also to anticipate to the potential problems that the presence of zoonotic parasites in fishery products may represent in the near future, as well as to look for solutions that allow the sector to be involved in its mitigation.
The first step is to assess the risk of the consumers to be exposed to zoonotic parasites. To do this, we carry out a comprehensive surveillance study on potentially harmful parasites in the most important food fish, such as Atlantic mackerel, the herring, hake, and anchovy, from all the major European fishing grounds, including the North Sea, the Baltic Sea, the Grand Sole Bank, and the Mediterranean. Additionally, we look at potentially harmful parasites in some common fresh fish imports to Europe. At the end, the surveillance study will result in a complete set of data ready for quantitative risk analysis.
The high sample effort conducted within the project has been reinforced through the development of a biobank, an advanced biorepository that stores biological samples, assuring efficient management with complete traceability in serving exchange of data and samples. This innovative tool, with 4 active nodes in Vigo, Bergen, Rome, and Madrid, will facilitate future multidisciplinary studies which will be able to use the samples maintained in the biobank. Moreover, the information gained will be used for developing epidemiological maps that will include information on, for example, demographic infection values.
Crucial point in the epidemiological survey carried out on the fish species target of this project is just the identification of the parasites recovered. Indeed, it is important to distinguish zoonotic parasites from those whose potential infection role to humans has not yet been well known or clarified. Thus, this part of a project is dealing just with the genetically identification of a huge amount of parasites coming from the biobank of the project, and the parasites are characterized based on their DNA analysis. In addition, an innovative methodological approach based on PCR DNA technology was developed in order to detect parasites and their fragments just directly in fish fillets and seafood products.
Although it is well known that anisakis is a fish parasite that can cause allergic reaction in humans, the diagnostic value of the test used to evaluate the anisakis allergy is limited by its low specificity. With Within the PARASITE Project, we have characterized the human immune responses to anisakis and identified 28 new parasite allergens, which are the proteins that may trigger an allergic reaction. This will contribute to the development of more specific diagnostic methods and improve treatments through personalizing diets, for example.
One critical step is the detection of parasites in fish samples. Within the PARASITE project, some detection methods have been improved, mainly the UV press method as an alternative to traditional, less efficient ones. This technique uses the fluorescence of deep frozen anisakid larvae, and it is based on visual inspection of pressed and subsequently deep frozen fillets under UV light. It can detect all anisakids in the edible part of all fresh or frozen fish species, as well as in the viscera and in some processed products like marinated or salted herring or anchovy fillets. For other kinds of processed fishery products like minced or heat-treated products, for instance canned or surimi, alternative detection methods based on molecular or immunological approaches are needed, which are being improved within the project as well.
If anisakids larvae are present in the fish, the risks they pose for consumers have to be reduced. The ways to minimize these risks have been studied and improved within the PARASITE project. Therefore, treatments for inactivation of anisakids larvae in fishery products have been optimized. New methods for antigen elimination or inactivation have been developed, and also a device to kill anisakis larvae in fish offals on board has been enhanced. These proposed interventions all along the value chain could contribute to reduce the risk for consumers, including sensitized patients.
With all this information gathered is to put it together so that we can predict the instance of infection and allergy in the human population in Europe. So to do that, what we carried out is a quantitative risk assessment.